episode of BioLogic Science News. Today I want to talk about golden rice and its latest incarnation in the world of agricultural biotechnology. In Eastern Asia, rice is a staple food. It's eaten with virtually every meal. It's a huge part of everyone's diet. Rice is generally pretty good for you too, but it's not like it's loaded with nutrients. It's just a decent carbohydrate that serves as a base food in the diet of more than 1 billion people. When researchers tried to address the growing problem of vitamin A deficiency in Asia, which can cause blindness in children and young adults, they turned to rice as a vehicle for carrying vitamin A. This led to the development of golden rice, a genetically modified species of rice that produces beta-carotene, which gets converted in the body into vitamin A. This would become known as the original golden rice, or golden rice 1 or 1.0. But there was a problem. The amount of beta-carotene produced by golden rice 1.0 was pretty small, too little to effectively treat vitamin A deficiency. By 2005, researchers had developed golden rice 2.0, which had more than 23 times the amount of carotenoid compounds than the original strain. Now golden rice is an effective dietary treatment for vitamin A deficiency. But then the researchers asked, why stop here? Why not try to create rice loaded with other key vitamins and nutrients? This led to the development of a range of rice strains, some of which were high in zinc, others that were high in iron, others that were high in this nutrient or that vitamin, etc., etc. This pattern of rice specialization may seem like a great development, but researchers reasoned that it kind of defeated the purpose. If you want people with chronic nutrient deficiencies to get the nutrients they need, you don't want them to have to buy 24 different kinds of rice. Preferably, you'd want a single rice strain with everything that you want in it. This was the goal of researchers at the ETH, the Eidgenössische Technische Hochschule, or the Swiss Federal Institute of Technology. To do this, they needed to really play with the stuff of life. They had to take genes from multiple different species and transplant them into the rice genome in a way that would work as intended. For vitamin A, they took a gene from a species of corn. This gene worked near the end of a biochemical pathway that produces beta-carotene in the corn plant. The reality is that rice naturally has this pathway, albeit in an immature form. The rice plant doesn't have all the enzymes it needs to finish the pathway and produce beta-carotene, but this genetic implant from the corn provides those missing enzymes, and it completes the otherwise immature biochemical pathway. And the rice can now produce beta-carotene. When we ingest beta-carotene, it gets converted into vitamin A. For zinc, they took a gene from a genus of plant called Arabidopsis. This is a small, fast-growing plant that's often used as a model organism. It's really useful for studying plant growth and genetics because it grows rapidly and its simple body design allows scientists to easily identify the changes caused by a gene alteration. The gene in question is nicotinamine synthase 1, which produces chemicals called metal chelators, and these bond with zinc ions. They also bind iron atoms, but they used a different protein specifically for iron atoms. This gene was taken from a bean plant. It produces a protein called ferritin that acts like a giant bag. A single protein of ferritin, just one big enzymatic bag, can capture and hold more than 4,000 iron ions, which are pretty big and heavy ions on their own. So that many of them, that's huge on a molecular level. All of these enzymes are linked together in the genome so that they all get expressed at the same time. Together, they allow the rice to produce safe levels of iron, zinc, and beta-carotene, which serves a really useful and healthy function in populations with these particular nutrient deficiencies. The catch is that while golden rice 3.0 has zinc and iron, it has way less beta-carotene than golden rice 2.0. A gram of 2.0 rice had around 24 to 30 micrograms of beta-carotene, while a gram of 3.0 rice only has 3.3 to 3.5 micrograms of it. So that's like 8 times as less, give or take, 8 or 9 times as less. This means the researchers at the Swiss Federal Institute of Technology have their work cut out for them. They're now trying to reach 4 micrograms per gram as a realistic goal, with more optimistic goals of a beta-carotene content between 8 and 12 micrograms per gram of rice. Hopefully they can do it, and hopefully the fruits of their labor will make it onto the market in China and the rest of Southeast Asia, where they can help huge numbers of people currently suffering from chronic deficiencies in vitamin A, iron, or zinc.